Good afternoon, everyone, and thank you for joining us today to both recognize and celebrate our 2021 Mayo Clinic Summer Undergraduate Research Fellows. My name is Leanne Carson, and I'm the Clinical Partners Manager at the Interdisciplinary Health Sciences Institute, and I coordinate the Mayo Clinic and Illinois Alliance education programs on the Illinois campus. Each year, Mayo Clinic invites around 180 undergraduates from across the nation to participate in their Summer Undergraduate Research Fellowship Program, or SURF. Sponsored by the Mayo Clinic Graduate School of Biomedical Sciences, this 10-week program gives undergraduates a chance to work at the forefront of biomedical research at one of the world's leading medical centers. Students work in the laboratories of Mayo Clinic faculty on standalone research projects or as part of ongoing investigations. They develop their technical skills, attend weekly seminars, and test their inclination toward a career in biomedical research. As part of our long-standing collaboration through the Alliance, Mayo Clinic reserves 7 spots each year for University of Illinois Urbana-Champaign students who are chosen by the Mayo Clinic and Illinois Alliance pre-selection committee. Because students are selected for their innovative excellence, often more than 7 U of I students are invited to participate in this prestigious undergraduate fellowship. This year, 11 outstanding students were selected to participate in the SURF program. We have asked each of our fellows to prepare a short video presentation of their project. After each video, I will ask the fellow to turn on their camera and microphone to share some brief remarks about their SURF experience. While we don't have time for questions today, we do invite those of you attending to use the chat feature to provide messages of appreciation and congratulations to the fellows. We ask that you keep your camera and microphone off during the student presentations. And we are recording the event today, and you will be able to access the recording on the Mayo Clinic and Illinois Alliance website following the event. Let's get started by taking a quick look at some of the experiences our fellows had this summer.
Hey everyone, I'm Vincent Lamb.
Hi everyone, you're going to be following me around for the rest of the day.
Let's go do some science.
Another great thing about working at the Mayo Clinic is that you get to shadow doctors and physicians in the hospital. This is a picture of me from last night after shadowing in the pediatrics emergency department.
My first research internship was actually in immunology. So I've had an interest in immunology, virology, and related fields for a while. But this interest has only deepened since the COVID-19 pandemic has started. It's interesting conducting virology research during a pandemic caused by a virus. A lot of the techniques I use to study HIV here at Mayo Clinic and on the Illinois campus can be applied to studying COVID-19. This makes me realize how relevant my work is and fills me with even more passion to pursue this field in the future.
Yes, indeed, the fellows did make us proud, and now we're going to hear from each of our 2021 fellows. Our first fellow is Neha Arun. This summer, Neha worked in the lab of Dr. Andrew Badley, and her research focused on how trail short is associated with human T cell activation.
Hi everyone, my name is Neha Arun. I'm a junior at UIUC studying MCB, and this past summer I worked in an HIV immunology lab under Dr. Andrew Badley with Dr. Sharzad Jalali. So my project last summer focused on a splice variant of trail called trail short and how it's associated with human T cell activation. So some background. The Badley lab discovered trail short in cells that were resistant to normal death, which we call apoptosis, and this was seen in HIV infected cells and cancer cells where you'd normally want the cells to die, but somehow this splice variant trail short was preventing them from doing that. So my hypothesis for the project was that trail short is associated with human T cell activation, which promotes resistance to trail-mediated killing. So you can see my methods here. They're all various types of flow cytometry assays. So this figure demonstrates that as we added decreasing concentrations of trail short in comparison to this red line here, which is the control, there is increasing amount of activation. So the more calcium that is released, the more activation we can infer. This idea is further reinforced using a cell line model, where if trail short cannot bind to a certain receptor, it can't release calcium. And then when this ionomycin is added, which releases all the calcium in the cell essentially, the full amount of calcium is released. However, if it does have the receptor it can bind to, trail short causes an initial release of calcium. So later on, there's actually less to be released. So this further reinforces this idea. We also stained for markers, CD25 and CD69, which show activation, and you can see that there is a percentage increase from the control to the trail short treated cells. We wanted to further enforce this idea by looking at proliferation, because proliferation is driven by activation. And once again, you can see that trail short coated wells had 7.9% more proliferating cells. So what does this mean? We can see that trail short facilitates a release of calcium, which is an indicator of T cell activation. And this idea was reinforced by the viewing of increased proliferation as well. So therefore, we can conclude that trail short is associated with T cell activation, and therefore its role in calcium signaling pathways and human T cell biology needs to be further understood so we can work on these issues such as HIV and cancer and other pressing problems. Here are my references and acknowledgements, and thank you so much for this opportunity.
Thank you, Neha. And now I'd like to invite you to turn on your camera and unmute and share any additional thoughts you might have.
Yeah, thank you again for this opportunity. A quick thought I just wanted to share is that my experience at Illinois has really prepared me for this opportunity. As I mentioned, I study HIV here on campus, and getting to do that at Mayo Clinic was a really great experience. And I can't wait to keep building this connection between U of I and Mayo for future students that may be interested in the program.
Thank you. Appreciate you sharing that. And congratulations on a great summer. Our next fellow is Evan Dre. This summer, Evan worked with Dr. David Daniels in the Department of Neurologic Surgery, studying pediatric brain tumors.
Hi everyone, my name's Evan and I'm a senior majoring in chemistry and MCB. This summer, I worked with Dr. David Daniels in the Department of Neurologic Surgery, studying pediatric brain tumors. And more specifically, we looked at diffuse intrinsic pontine glioma, or DIPG. DIPG is an aggressive glioma of the brainstem that typically presents in children between the ages of 5 and 10. And it's truly a death sentence. The median survival is less than 6 months. And once you get to about a year post-diagnosis, less than 1% of children are still alive. On the left, we can see an MRI scan of an 8-year-old girl who was diagnosed with DIPG at the Mayo Clinic a few years back, who had agreed to participate in research. And you can see the arrow pointing to the mass in the middle of her brainstem. And so this raises the question, what can we do about it? What are our treatment options? The first is surgery. Now, with this particular tumor, surgery is not an option because of the anatomic location. If you were to cut out the pons, you'd be doing more harm than good. The second option is radiation, which is simply palliative. It doesn't extend their life in any way, and it's certainly no cure. And so that leaves us with chemotherapy, which for this particular tumor also isn't an option because this tumor leaves the blood-brain barrier intact. And so with an intact blood-brain barrier, intravenous drugs can't get into the brain parenchyma, they can't traffic to the site of the tumor, and they can't have their cytotoxic effects. Now, a lab a couple of years ago had shown that vesicles, extracellular vesicles, are able to bypass the blood-brain barrier and enter the brain parenchyma. And that's of interest to biologists like us that are trying to find ways to get drugs into the brain. And so we asked the question, can we load vesicle with drug? And is drug-loaded vesicle an efficient way to potentially treat this tumor? And so in our first set of experiments, we show that with 5 different drugs, we can efficiently and effectively load the vesicle with drug. The next question we asked is, in vitro, can DIPG cells take up vesicle? Shown in blue are the cancer cells, and then shown in green is fluorescently labeled extracellular vesicle. And so we can see over time, the DIPG cells do take up the drug or the vesicle intracellularly. The next question we asked is drug-loaded vesicle able to induce cytotoxicity in DIPG cells in vitro? Shown in red is drug-loaded vesicle, shown in black is drug by itself. And we see that the drug-loaded vesicle is able to induce cytotoxicity to an even greater degree than drug by itself. And finally, we used a mouse model of DIPG. So it had a human-derived DIPG tumor within the mouse brain. We then did an IV infusion of vesicle that's shown in green, the tumor is shown in red. After about an hour, we sacrificed the animal, cryosectioned the brain, and imaged it under a microscope. And we can see that the vesicle did bypass the blood-brain barrier. It entered the brain parenchyma, trafficked to the site of the tumor, and we can see that in the merged image on the right. And so overall, we showed that we were able to effectively load vesicle with drug. The drug-loaded vesicle was able to induce cytotoxicity, and it was able to pass the blood-brain barrier and enter the site of the tumor in a mouse model of DIPG.
Thank you, Evan. I'd now like to invite you to unmute and add any comments about your SERP experience.
Yeah, I just wanted to thank the Illinois and Mayo Clinic Alliance for this opportunity. I've never worked with a group of people so passionate and dedicated about what they do, and it was truly inspiring, and I learned a lot. So, yeah.
Wonderful, thank you, and congratulations on having a great summer at Mayo. And our next fellow is Edward Huang. Edward worked with Dr. Adrienne Ting, who works with tumor necrosis factor and its potential as a revolutionary immunotherapy for cancer treatment.
Hello everyone, my name is Edward and I worked in Ting Lab this summer and I studied the sensitizing effects of TRAF2 deficiency on TNF and IFN gamma induced cancer cell death. To set the context for my project, I worked on novel ways of improving cancer immunotherapies. So currently, the 2 major checkpoint inhibitor immunotherapies we have are anti-PD-1, which is nivolumab, and anti-CTLA-4, which is ipilimumab. And this is a figure I pulled from a study in 2019 that shows in human patients with melanoma, after 5 years, there's still about a 52% survival rate even if we give them a combination treatment of both of the checkpoint inhibitors. And so this is significant, but there's still a lot of room for improvement. And so under that context, I studied tumor necrosis factor and interferon gamma, which are 2 cytokines in the immune system that have a lot of functions, but I decided to focus on their function in cell signaling, specifically cancer cell signaling. And so TNF is able to activate both a survival pathway and a death pathway in cancer cells. Unfortunately, the default pathway for TNF is the survival pathway. And so my project focused on ways that we could kind of flip the switch and what proteins we can mess with to switch it over to the death pathway, which induces apoptosis from within the cancer cell and killing it that way, rather than bombarding it with stronger and stronger weapons from the outside immune system, which has been the traditional method of improving cancer immunotherapies. And so I decided to examine TRAF2, which is a protein along the pathway that has been indicated, a knockout has been indicated to induce more cell death in cancer cells. However, no one has actually looked at how a combination treatment of TNF and IFN gamma, what it would do to a TRAF2 knockout cell. And so I began with a line of mouse melanoma B16F1 cells, and I did a CRISPR-Cas9 knockout. And you can see here in the Western that there's still a little small remnant of TRAF2. And so I went ahead and did single cell cloning, and I got these much cleaner knockouts we can see in this Western here. And then I pulled these colonies into one complete colony, which I called the TRAF2 knockout complete colony. And so the main experiment I ran was an IncuCyte cell death assay, and I would put my cells into these wells on this plate, and each well would have a different cytokine treatment. And then I'd throw it into this machine, which would track cell death and cell growth. And so here are the graphs from that. And we can see for just the cell death, we can see the single treatment of TNF alone does show significant death in the TRAF2 knockout cells compared to the wild type. The single treatment of IFN gamma doesn't really show any cell death, which kind of indicates a cytostatic effect, which is what we expected. And then a combination treatment of both cytokines shows also significant cell death in the TRAF2 knockout cells compared to the wild type cells, although not much different from just the TNF separate treatment. And then when we look at cell growth, both the individual treatments show about the same trend, and then the combination treatment shows that there is actually inhibited cell growth with the combination treatment. And so the IncuCyte machine gave me a metric for cell death and gave me a metric for cell growth. But as you can see, it seems like there's an incomplete picture here, and I really wanted to get a more clear picture of the situation. So I created the net death index, which is, it takes the number of cells that are number of dead cells and normalize it to 1% confluence on the area of the cell culture plate. And then when we do that, we can see the net death, and now it becomes a lot more apparent that the TRAF2 knockout cells are dying a lot more compared to the wild type cells, compared to any of the separate treatments alone of the other cytokines. So in summary, TNF doesn't really show killing in wild-type cancer cells, and it shows increased killing in TRAF2 knockout cells. IFN gamma inhibits wild-type cancer cell growth slightly, but shows significant inhibition of proliferation in TRAF2 knockout cancer cells, especially in the combination treatment. Thank you.
Due to teaching obligations, Edward is unable to join us today to provide any remarks, but we do want to congratulate Edward on his work this summer. As you can tell, all of these students do significant, they're outstanding students who do great projects over the summer. So, so congratulations to Edward as well. And our next fellow is Victoria Kindratenko. And Victoria worked in Dr. Alexander Revzin's lab and focused on organ-on-chip models for drug testing applications.
Everyone, thank you all for joining us and learning more about our summer experiences at Mayo. My name is Victoria Kindratenko, and I am a junior in bioengineering. And this summer, I worked in Dr. Alexander Revzin's lab, and I was mentored by Manolo de Hoyos and Dr. Alexander Revzin. So my research focused on developing organ-on-chip models for drug testing applications. And the way this model works is that it fluorescently tracks a protein that indicates tissue viability and functionality produced by a certain tissue. So if you insert a gene for a fluorescent protein before the viability protein gene, you'll produce the fluorescent protein when you produce the actual protein. And this fluorescence allows for the continuous real-time visual tracking of tissue viability, and you don't have to perform any complex assays or anything like that. So this summer, I focused on the first step of developing this kind of platform. And the first step is getting the fluorescent protein gene sequence into the DNA of the tissue. This gene insertion can be achieved with the use of a lentivirus vector, otherwise known as transduction. These viruses infect cells by integrating their own genome into the hosts, which allows for long-lasting gene expression. However, studies have only been able to successfully transduce thin hippocampal slices, and few explore transduction in liver and kidney samples prompting the need for an optimized liver and kidney transduction protocol. And also, few publications discuss transduction in microfluidic devices. Microfluidics are a novel approach to transducing tissue. They have a variety of benefits, including confinement of samples into small volumes, which reduces the amount of reagents required and increases diffusion rates into the sample. So to conduct these experiments, we collected needle core biopsies from rat, liver, and kidney tissue, as shown here. And we injected it into these little devices. They're about the size of a dollar coin. And then we injected media with lentivirus into the wells of the device and incubated for 24 hours. Then we removed the media and replaced it every 24 hours and collected it. We also used the LDH cytotoxicity fluorometric assay to measure the level of cellular damage in the kidney tissue. So we did a couple of preliminary trials and we compared two needle gauge thicknesses and saw that the thicker needle gauge was viable for longer. And we also saw that we were not able to transduce the liver tissue. So we continued our experiments with thicker kidney samples. Imaging showed bright transduced nuclei as shown here. And this meant that the virus had been able to enter the cells. Since the virus was able to enter, we knew that we should expect larger cell damage levels, which we measured with the LDH assay. And LDH fluorescence signal is directly proportional to cell damage. So figure 4A shows the tissue transduced with the lentivirus had increased levels of LDH activity in the higher cytotoxicity. So we then used a cryostat to perpendicularly slice the tissues just to see the depth of viral penetration. And as you can kind of see in the top right, the cells only on the exterior were able to be transduced, not a lot on the interior, not a lot of red fluorescence there. So this may be due to the lower virus concentration or just the fact that the cells on the interior were not easily accessible. So the preliminary and final results indicate that further research is warranted in increasing transduction efficiency. To develop a robust and effective organ-on-chip model, cells on the interior tissue samples must be monitored as well. Detergents such as polybrine, dextran, and collagenase, or electroporation are potential methods for improving transduction. These detergents will loosen the cell-to-cell connections and potentially improve the depth of viral transduction. So increasing the concentration of virus in the microfluidic devices is also a potential solution. Low cytotoxicity is required for preparing higher titer lentivirus concentrations to improve transduction efficiency though. So overall, we had success in transducing the kidney tissue, not the liver tissue, and it could be significantly improved still. And continuing research down this path will help create personalized gene therapies. So I'd like to thank everyone in the Revson Lab and the Mayo Illinois Alliance for making all this possible. Overall, I had a great time in Rochester, Minnesota, and I enjoyed meeting other SURF students and exploring Minnesota. I also love working in my lab. The other SURF students and my mentor were fantastic to work with. I look forward to seeing where my career takes me, and I hope it includes Mayo. Thank you.
Thank you, Victoria. Now we'd like to invite you to unmute and say a few remarks.
Hi, Leanne. Thank you. Thank you, everyone, for coming. I had a great time as a Mayo Surf student this summer, and one of the most useful and helpful classes here at UIUC that I took was Bioengineering 202, the cell tissue engineering lab, which is actually where I am right now. I'm in LA for one of the sections. So overall, great experience. And this lab is definitely very helpful in teaching me all the techniques that really prepared me for my research during the summer in Rochester. Thank you.
Thank you, Victoria, and congratulations on your summer. And thank you for participating in the bioengineering Instagram takeover. That was fun. So thank you. Our next fellow is Vincent Lamb, and Vincent also participated in the bioengineering Instagram takeover. So, and Vincent Vincent worked with Dr. Quinn Peterson this summer in regenerative sciences to model mitochondrial disease using iPS cells derived from MELAS patients. So let's hear from Vincent.
Hello, everyone. My name is Vincent Lamb, and I'm a junior in bioengineering. This summer, I worked under the mentorship of Dr. Zenith Kashim and my PI, Dr. Quinn Peterson, in the Department of Regenerative Sciences. The title of my project is Modeling Mitochondrial Disease Using iPS Cells Derived from MELAS Patients. To provide some background information, MELAS stands for mitochondrial encephalomyopathy, lactic acidosis, and stroke-like episodes. This disease affects many organ systems, but it can also lead to endocrine disorders such as diabetes. In addition, heteroplasmy is a mitochondrial disease marked by the presence of multiple mitochondrial genomes. When used in conjunction with other mitochondrial diseases, heteroplasmy can indicate the level of severity. Due to the lack of existing literature on beta cell differentiation regarding MELAS patient-derived induced pluripotent stem cells, this experiment would be very beneficial to modeling how MELAS plays a role in endocrine disorders. The objective of my research this summer was to study how heteroplasmy in MELAS iPSCs affects the beta cell differentiation and behavior. I subjected different heteroplasmic clone lines of iPSCs from a MELAS patient through a stem cell-derived beta cell differentiation protocol developed by the Islet Regeneration Laboratory. Upon completion of each stage of the differentiation protocol, I took samples for flow cytometry to analyze the cells for the correct biomarkers. As you can see in these results, all 3 clones expressed expected levels of biomarkers up to stage 3, and all 3 clones expressed chromogranin, an endocrine-specific biomarker at stage 6, day 6. However, at stage 6, day 10, co-positive insulin and NKX6.1 expression was minimally observed in all clone lines. This experiment proves that MELAS iPSCs can differentiate into beta cells in 2D cell culture, and the percentage of cells per stage appeared to not be directly related to heteroplasmy levels. The reduced NKX6.1 expression may be an indicator of non-functional beta cells, which could be caused by differentiation in 2D cell culture or due to the mitochondrial dysfunction of MELAS cells. In addition, insulin expression in stage 6, day 10 cells was lower than expected, which will limit their insulin response to glucose. Overall, the data suggests that heteroplasmy does not affect beta cell differentiation of iPSCs derived from MELAS patients. If I were to continue this experiment, I would like to run a glucose-stimulated insulin secretion assay to assess the effects of heteroplasmy on beta cell functionality. I also compared the differentiation process of MELAS iPSCs in 2D and 3D cell culture. I'd like to thank the Mayo Clinic and Illinois Alliance so much for this amazing opportunity to meet world-renowned faculty, scholars, doctors, and exceptional students from across other institutions. I'm so grateful for how this experience has helped me prepare for a future in medical research, and I would definitely recommend this program.
Thank you, Vincent. I'd now like to invite you to unmute and share your thoughts with us.
Sure. Hi, everyone. I like to say that this program has really been beneficial to deciding whether I want to pursue an MD PhD program. Obviously, in this program, you get to meet a lot of medical and graduate students, and they were just talking with them. They just provided their insight into the application process and just their life as a student. And definitely, I am looking forward to applying to Mayo Clinic in the future.
Thank you. Appreciate that. And congratulations on a great summer.
Thank you.
Our next fellow is Sarah Madatov. This summer, Sarah worked with Dr. Marissa Schaefer, studying the link between lung injury, senescence, and brain dysfunction.
Hi, everyone. My name is Sarah Madatov. I'm a senior in biochemistry and psychology, and this summer I did work in Dr. Marissa Schaefer's lab studying the link between lung injury and senescence and brain dysfunction. So cellular senescence is a really wide research field that's currently being studied in relation to aging. Cellular senescence is basically when cells get DNA damage, and instead of dying or becoming cancer cells, they stop dividing and start secreting inflammatory markers into their local environment and also into the periphery. Most of the time, these senescent cells show up as we age, which is why they're implicated in a lot of aging diseases and causing a lot of inflammation that comes with age, but they also occur after injury. We wanted to look specifically at the lungs because there's been a lot of studies recently that link lung injury with brain dysfunction, and this link cannot be explained by just a loss of oxygen input into the body through less functioning lungs. So our project had 2 specific aims. First, to establish a link between lung injury and increased brain expression of inflammatory and senescent markers in both young and old mice, and also to investigate the production of inflammatory markers in fibroblasts after they're injured with bleomycin, which causes them to become senescent. Our experiment had 5 different groups. So we had a young mouse group that was treated with only PBS, a young mouse group that was treated with bleomycin, and then allowed to recover for 30 days, which is usually the amount of time that it takes for these mice to recover from their lung injury. We wanted to see if recovering from their lung injury still showed effects in the brain. And we had 3 conditions for the old mice. So we had old mice that were just treated with PBS, and old mice that were treated with bleomycin. Some were allowed to recover for 30 days, and some were allowed to recover for 60 days, just to see if maybe it takes old mice a little longer to recover. We tested the brain inflammatory markers in 3 different brain regions: the cerebellum, the hippocampus, and the hypothalamus. And in all 3, we saw a significant increase in both senescent markers and inflammatory markers, both based on age distinction and on bleomycin treatment distinction. So in the fibroblasts, we also were able to see a really significant increase in inflammatory marker expression, which kind of shows us that probably these fibroblasts, once they're injured and become senescent in the lungs, they start creating and secreting inflammatory markers, which then make their way to the brain and cause the brain to increase its production of inflammatory and senescent markers, and maybe causing this brain dysfunction. Results of this study are really important in beginning to understand aging as a systemic interaction, where injury in one organ may drive injury in another organ, or do things like accelerate aging in the brain. In the future, this study is going to be explored further to find a more fundamental link between the lung injury and the brain injury by injecting the inflammatory markers that are actually secreted by these fibroblasts. When they were in culture, we collected the conditioned media. This conditioned media will both be injected into mice to see if we can just cause brain dysfunction based on only this media being injected. And also it'll be characterized using a multiplex assay to see what inflammatory markers are actually present inside. Once we have this final link into the project, you can really see how the lung injury can be very nicely linked to the brain dysfunction that we see. And that'll be really interesting going forward into the field of brain dysfunction, where usually if you are studying the brain, you only study the brain. But here we show that maybe what's happening in the brain is caused by another part of the body.
Thank you, Sarah. Unfortunately, Sarah is not able to join us today. She also has teaching obligations, but we do want to congratulate Sarah on her important work this summer. And our next fellow is Ege Onal. Ege worked in the lab of Dr. Michael Romero to study kidney stone formation using a fruit fly model.
Hi, everyone. My name is Ege, and over the summer, I work with kidney stones. So kidney stones are these rock-like structures that form when the urine becomes supersaturated with organic or inorganic materials. And as you can imagine, the formation of kidney stones is a painful process, and it damages our kidneys. So in order to study kidney stones, in this study, we used fruit flies and observed crystal formation. In a previous canine study, it was seen that anactiniform expression has been associated with calcium oxalate stone formation. And in a separate human study, it was seen that the urine samples of calcium stone formers had relatively lower levels of anactinophore markers on the extracellular vesicles compared with the non-calcium stone formers. So this leads us to ask, does anactinophore expression affect calcium oxalate kidney stone formation? To study calcium oxalate stone formation, we won't be able to use humans or animals. However, the fruit flies are an amazing model because we can easily do gene knockdowns, and the Malpighian tubules of fruit flies are functionally homologous to human kidneys, and they do in fact form crystals. So the closest genetic ortholog of anactinin-4 in fruit flies is subdued. So this is particularly interesting because subdued in a separate study has been seen that had a protective role against gram-negative bacteria. And the researchers saw that knockout flies had lower percentage levels of survival compared with the wild type. And historically, we also know that urinary tract infections are associated with kidney stones. So this leads to our hypothesis. Subdued knockdown leads to more crystals, and the question is there a role of bacterial infection? So in order to do gene knockdowns, we use the Gal4-UAS system. In our first fly, we have the urate oxidase tissue-specific promoter, allowing us to target the principal cells of the Malpighian tubules, and we have our Gal4 transcription factor. In our second fly, we have our upstream activation sequence and our subdued RNAi. The F1 generation of flies have efficient mRNA degradation and gene knockdown. So in our study, we had two groups, knockdown and control. And to both of these groups, we fed sodium oxalate with and without EGFP tagged gram-negative bacteria. After 4 days, we dissected them, and here's a video of me doing one of the dissections. So now this is the back of the fly. I'm taking out the gut. And the first tubule that just got out is the posterior tubule. The second tubule that got out now is the anterior tubule. In our study, we used the anterior tubule as a control, and also since the anterior tubule is closest to where the nutrients are being taken up from. So now I'm isolating the anterior tubule from the rest of the gut by cutting from the ureter. After isolating, I basically put them on polyolysine slides and imaged them using polarized light microscopy. So the results of the sodium oxalate feeding quantitatively show that the crystal counts between subdued knockdown and subdued control groups were relatively similar. However, the crystal size of subdued knockdown was larger compared with the control. And in fact, once we look at the pictures, we can see that the knockdown in fact has larger crystals. Our sodium oxalate plus UPEC feeding experiment showed that the crystal count and crystal sizes between the subdued knockdown and control are quantitatively not different. However, the images tell a different story. In the images, we can see that the knockdown, in fact, has aggregates of crystals being formed. And we can take a closer look and see that these aggregates, in fact, crystals have similar sizes with the control, but they formed aggregates of crystals in specific regions of the Malpighian tubules. So as a conclusion, larger crystals form in subdued knockdown versus control. UPEC leads to aggregation of crystals in subdued knockdown, and inactioniformis expression is likely causative of kidney stones, yet we do not know the exact mechanism yet. I would like to thank my principal investigator, Dr. Ramiro, the NUSERV program, Dr. Reynolds, Danny, Heather, and Mayo Clinic and Illinois Alliance for making this a dream come through. And I would like to thank all the amazing and wonderful people I met in this program that have made this a really special experience and opportunity. Thank you.
Thank you, Ege. Would you like to unmute and add a few comments?
Yes, absolutely. Thank you. So I would like to take this opportunity to thank the Mayo and Illinois Alliance for giving this really special opportunity to grow as a scholar. At Mayo Clinic, having Dr. Ramiro as my mentor in a hands-on cutting-edge research project really allowed me to widen my horizon about biomedical research and led me to understand what it truly means to be a researcher. Through my experiences, I learned that Mayo Clinic isn't only a pioneer in medical research, but it's an institution that fosters a community of growth and mentorship with invaluable mentors such as Dr. Ramiro, Dr. Horozdowski, Dr. Wenk, Kit Knayer, Dilip Moni, Ramin Jarmani, and their guidance has been really eye-opening for my future endeavors. Last, I want to say that I feel truly blessed to be a part of this amazing institution and brilliant individuals. Thank you very much for this opportunity.
You're welcome, and thank you for your kind comments. And our next fellow is Danny Owen. Danny worked with Dr. Lewis Roberts to understand the relationship between hepatocellular carcinoma biomarkers and the genomic constitution of the tumors.
Hello and welcome to the Illinois Mayo Clinic SERF celebration. I'm so excited to walk you through my project and talk about my experience. So my project was understanding the dynamics of HCC biomarkers in correlation to the genomic constitution of the individual tumors. So what is HCC? HCC is a type of liver cancer, and it's one of the leading causes of cancer deaths worldwide. There are a lot of common risk factors for HCC: hepatitis, alcohol use disorder, fatty liver disease. So patients with these risk factors are often screened for their biomarkers over time before they develop cancer so they can be caught earlier. So there's a lot of data out there of biomarkers over time, both before a patient is diagnosed and while a patient has HCC. So our primary objective was to understand the relationship between the dynamics of these biomarkers over time and the constitution of the tumors. So how do we go about doing this? We split the patients into 3 groups The first group had biomarkers that improved over time, the second group had biomarkers that worsened over time, and the third group had no clear pattern in their biomarkers. And we looked at their survival. So patients whose biomarkers improved over time had really great survival, and that represents the blue line here on this curve, whereas patients who had worsened biomarkers over time had a little bit worse survival. So with that survival data, we went and correlated it with the genomics of the individual patients, and we tried to identify genes that were common in the groups. So for example, in patients whose biomarkers improved, the DYNC2H1 gene was the most common and not present in any of the other groups, whereas patients with worsened biomarkers, an APOB gene mutation was the most common. But more significantly than that, we found that patients whose biomarkers improved over time had much fewer significant genetic mutations than patients whose biomarkers worsened over time. And you can see that in table 1. Patients whose biomarkers worsened over time, 270 genetic mutations on average, whereas patients whose biomarkers improved over time, much fewer at 6. So what does this mean? Well, it means that the genetic constitution of an individual patient's HCC may influence their progression and outcome. And this could lead to the development of individualized treatment plans for these individual patients that fit their needs specifically. It has been shown that, AFP response can be used to predict liver transplant outcomes. So the goal would be to further this research and see how it could improve liver treatment outcomes as well, liver cancer treatment outcomes. We found a lot of significant results with these genetics, but our sample size were really small, so more research is warranted just to get rid of the possibility of a false discovery with these particular genetic mutations. I would, of course, like to thank Dr. Roberts and Dr. Muhammad Ali for mentoring me throughout the course of this summer and letting me participate in this research project. I would also like to thank the University of Illinois for sending me to the Mayo Clinic and the Mayo Clinic for having me. It was such a wonderful experience to get involved with clinical research. It was my first time, and now I'm much more certain that this is the direction I want to go with my career. I would also like to thank all of the friends that I made in Rochester, a lot of them Illinois students. I was able to broaden my friendships with them, and I know that these friendships are going to last a lifetime. So thank you.
Thank you, Danny. And I would like to invite you to unmute and add any comments.
Yeah, absolutely. So thank you. I would first like to thank the University of Illinois for preparing me for this experience. I've actually been doing basic liver cancer research here for around 3 years, and I felt well prepared for this experience. So I would like to thank my PI here for that. I would also like to thank Mayo for taking me and pairing me with such a great mentor. I learned so much, and being able to integrate the human experience of cancer into my experience with cancer research at such a special patient-centric institution truly helped me determine the future of my career. So I would like to thank both institutions and the Alliance.
Thank you, Danny. Our next fellow is Vangai Tizora. This summer, Vangai worked in the skeletal development and regeneration lab of Dr. Jennifer Westendorf to identify different molecular targets of FLIP in chondrocytes.
Hi everyone, my name is Vangai and I'm currently a senior studying bioengineering here at UIUC. This summer at Mayo, I was in Dr. Jennifer Westendorf's skeletal development and regeneration lab, and I'm excited to tell you a little bit about what I was doing. FLIP stands for this really long acronym that I'm not going to take the time to explain, but basically it's a protein phosphatase. And what that means is that it removes phosphate groups off of other proteins or dephosphorylates proteins. FLIP1 in particular plays an important role in the development and regeneration of cartilage, and this has downstream implications for bone growth and also the progression of diseases such as osteoarthritis. The main purpose of this figure here is to illustrate that FLIP is involved in many different important cell signaling pathways like the AKT, PKC pathways, and as such that makes it a very attractive target for therapeutic intervention. We've seen in the past that when FLIP is inhibited, this leads to the increase of chondrocyte proliferation and differentiation. Chondrocytes are the cells that make up cartilage. One tool that we have at our disposal to help inhibit FLIP is called 11779. It is a small molecule, and we hypothesize that by inhibiting FLIP, that is what's leading to the progression through the cell cycle and may even explain some of the things that we're seeing in terms of increased chondrocyte proliferation. And that brings us to my research goal, which is to identify different molecular targets of FLIP in chondrocytes. Before I arrived at the lab, they basically ran this huge phosphorylation microarray looking at over 1,000 different potential candidates. And we received this huge list comparing the protein lysates produced from both wild type and FLIP1 knockout chondrocytes. So what I was doing was trying to validate the results that we received from the past through the use of Western blots. So instead of using primary chondrocytes, I was using ATDC5 cells, which is a chondrogenic mouse cell line. Given the potential candidates that we found to be of interest, we treated the ATDC5 cells with varying levels of the FLIP inhibitor 11779, and the results are summarized here. In conclusion, FLIP inhibitors may affect protein phosphorylation differently than what we're seeing with the germline deletion of FLIP1. And all this just goes to show that further studies are needed to further figure out what's going on in terms of FLIP inhibition and protein phosphorylation. At this time, I would like to thank all of the generous funding sources that helped make my research experience possible this past summer. In particular, I'd like to thank my PI, Dr. Jennifer Westendorf, and my mentor, Dr. Sam Weaver, for all of the careful guidance and support that they gave me this past summer. I'm so incredibly thankful for the opportunity that I had to put some of the skills I learned in the classroom to good use. And I know that the relationships and friendships that I made over my time at Rochester will last me a lifetime. Thank you.
Thank you, Vangai. And now I'd like to invite you to unmute and add any comments you might have.
Yes, of course. Hello, everyone. So first off, I just wanted to say, yes, I was extremely thankful for this amazing opportunity to grow more as a scientist. In particular, I would say I was pleasantly surprised with the amount of networking opportunities that we SURF students had. For one, my PI was aware that one of my goals is to become a physician and work within the health equity space. So she actually connected me with a physician at Mayo who was doing research in that sector. And I was able to gain a better understanding of what that might look like when I pursue medicine in the future. And in addition to that, I also participated in the new SURF program, which I'm wearing the merch for that right now. And that's basically a nephrology and neurology subset of the SURF program, because I am also interested in potentially becoming a nephrologist. So it was a great opportunity for me to learn a little bit more about the field and hear from experts within nephrology and neurology. So thank you again, and I'm hoping that Mayo will be in my future in some sort of shape or form.
Thank you. And it's great to hear about the additional opportunities that Mayo offers their SURF fellows. And we want to congratulate you on a great summer. And our next fellow is Angelina Wilton. This summer, Angelina worked with Dr. Arjun Threya, who is an Illinois alumnus, and Dr. Richard Weichelbaum on a project to characterize suicide attempts in adults with major depressive disorder.
Hello, everyone. My name is Angelina, and I'm a senior at U of I studying MCB and psychology. And I spent this past summer in the Athrea lab at Mayo Clinic working on a dry lab project characterizing suicide attempts in adults with MDD. So we wanted to see if we could differentiate patients with and without suicide attempt history in a sample of depressed adults using genomic and metabolomic markers. We conducted a systematic PubMed and GWAS catalog search to look for single nucleotide variants associated with suicide attempt. And after pulling those variants in our sample based on our search, that genomic and metabolomic data were integrated into a multi-omic network analysis using a package called XMWAS in R to assess relationships between genomic and metabolomic profiles in individuals with and without suicide attempt history. So you can see that in the networks on the top right. So we found that two variants, one in the clock gene and one in the BMAL1 gene, strongly differentiate both networks due to having a high delta centrality value, which is a statistical value that represents a node's relative influence between networks. So as you can see in the non-attempters, clock and BMAL1 are statistically correlated with several phosphatidylcholines, amino acids, and kynurenine, and these associations are absent in the attempter network. So it's well studied in the literature that clock and BMAL1 heterodimerize to regulate the transcription of circadian genes. And the relationship of these circadian metabolites with BMAL1 and clock regulated processes may be implicated or disrupted in suicide attempters, and maybe improving this understanding may help to more objectively identify individuals at an increased risk for attempting suicide. So along with working on this exciting project, I also participated in the Mayo Clinic entrepreneurship course, where I most importantly learned about what value actually is and how it applies to research. And I got to pitch our group idea Shark Tank style at the end, which was really scary and also really fun. So this summer was a lot of fun working with and getting to know my lab members and the faculty at the Department of Psychiatry at Mayo. So through a lot of coffee meetings and quite a bit of shadowing, I learned a little about what it means to be a physician, what it means to be a scientist, and what it means to be both. I want to give my thanks to Dr. Ethrea and the lab for giving me a wonderful experience and opportunity to learn. And I also had a lot of fun with the other Illinois surf students and surf students from across the country exploring Rochester. I wanted to thank the Mayo Clinic and Illinois Alliance for this wonderful and unforgettable opportunity.
Thank you, Angelina. Would you like to unmute and say a few things?
Yes, of course. So I want to reiterate my thank you to the Alliance and everyone who has mentored me along the way for this awesome experience, including Dr. Ethrea, Dr. Wenchelbaum, Dr. Paul Corkin, Caroline Grant, and also my PI on campus who prepared me for a intense research experience like this, Dr. Chung. I also wanted to thank the other Illinois surfs for a really fun summer. So this summer was my first time doing any sort of dry lab experiments. And through this experience, I was introduced to the strengths of computational and data science methods in the context of biomedical research. And I've been expanding my knowledge, taking some bioinformatics classes this year, which has been fun. And I will also be a poster presenter at the 10th annual Individualizing Medicine Conference this October. So I would love to discuss this project more in detail with any of you if you happen to be attending. So thank you for everything.
Well, congratulations on being a poster presenter. That's great news, and congratulations on a wonderful summer. And our final presentation this afternoon is from Andrew Zhang. This summer, Andrew worked with Dr. Isabel Scarisbrick to conduct research on myelin dynamics in a murine model of type 1 diabetes.
Hello, everybody. My name is Andrew Zhang. I was a surf student this summer in Dr. Isabel Scarisbrick lab in the Rochester campus in Mayo Clinic. And this past summer, I was conducting a research project on myelin dynamics, so myelin and cells that are associated with it, and seeing how they're affected by a mouse model with type 1 diabetes. So to give a quick background, first of all, myelin is really important in our nervous system. They basically wrap around axons. They have three main functions. The first is to propagate signals at a faster rate. The second is to prevent any decay of signals. And the third is to prevent multiple signals from interrupting with each other. And so basically, myelin is really important for just our neurons to tell our bodies what to do. And an issue is that there are many young adults and actually older population as well who are affected by demyelinating diseases. For example, multiple sclerosis, and this can cause really drastic lifestyle changes because of the damage of the neurons, therefore damage to the body. And how come we're looking at type 1 diabetes? Well, we already know that type 2 diabetes and Western style, like high fat diets, are already huge risk factors for the development onset of multiple sclerosis. And we were thinking that type 1 diabetes could potentially be the same. For example, oligodendrocytes, which are responsible for making the myelin in the central nervous system, and astrocytes, which are responsible for maintaining like the extracellular chemical environment of the brain, nourishing oligodendrocytes and neurons. Both of them have insulin receptors, therefore like the insulin hormone can actually interact and talk to these cells. And if in type 1 diabetes, there's less insulin, so that might affect the myelin phenotype. And so how do we do this? We basically had two groups of mice, one with the type 1 diabetes, another control. And we allowed the two groups of mice to eat like normal diets, just allow them to grow. And then we took histological images, So brain slices to see and stain if there's any changes in the protein or the cells in the diabetic mice compared to the control. And so what do we see? Well, for the oligodendrocytes that are responsible for making the myelin in the bottom left corner, there wasn't any changes from the control to the type 1 diabetic mice. However, we actually see a significant decrease in myelin binding protein in the diabetic mice, although we kind of see a reverse trend in proteolipid protein, which is also found in myelin, which could be indicating like dysfunction or damage in the myelin. And we also see that standing for GFAP, which is standing for astrocytes, there's actually a significant increase in astrocytes in the type 1 diabetic mouse model. And so our current hypothesis is that if there's like improper insulin hormone balance, that's improper signaling to the neural cells in the brain and the spinal cord. And what we see is we might see damage or malfunction of the myelin. And in order to at least mediate or rectify the situation, we see increased astrocytosis or the proliferation of astrocytes just to modify or mediate any effects. And so basically that's kind of my project this summer. I want to thank everybody again for giving us the opportunity to not only conduct research, but also share our work. It's an incredible opportunity, and I'm really appreciative of that.
Thank you, Andrew. Would you like to take this time to unmute and add a few comments?
Well, of course. Can you guys hear me?
Yes.
Okay, cool. I want to reiterate with a huge thank you to the University of Illinois and Mayo Clinic for this incredible opportunity. And a thank you to my PI, Dr. Isabel Scarisbrick, my mentor, Dr. Monica Langley, for helping me learn and grow this summer. I learned so much, even outside the research sphere. For example, one of the physicians I shadowed was a neurosurgeon, and I actually got to see the live human brain in person for the first time. So hands down, one of the best summers I had.
That sounds great. And congratulations on a great summer. All right, I know it's almost 5, but if you can just hang with us for a few more minutes, we're coming to the really fun part of this celebration. As a way to promote engagement among the SURF students and foster camaraderie, we held a SURF photo scavenger hunt this summer. We asked the students to take photos and to show their Illini pride at specific sites on the Mayo Clinic campus and in Rochester. And so you can see right here, the students are at the My Brother and I sculpture of the Mayo brothers. And so appearing in a photo earned an entry in our drawing and for a $50 gift certificate to the Illini Union Bookstore. So we're now going to draw for the winner of the gift certificate. I'm going to stop sharing and we're going to bring up our prize wheel.
Can you all see the prize wheel?
Yes.
Yay. Okay, we're gonna go ahead and spin it.
Yay. Congratulations, Victoria, you're the winner. So we will get you your $50 gift certificate to the Illini Union Bookstore soon.
Great, thank you.
Thank you. And I would like to close our celebration with a few special thank yous. Thank you to the members of the SURF pre-selection committee who have the difficult job of selecting our outstanding SURF students each year. And thank you to Dr. Bruce Horowsdowski, Glenda Mueller, and the Mayo Clinic Graduate School of Biomedical Sciences who went above and beyond to ensure our students could safely attend the program in person this year. So a big thank you there. Thank you to the Mayo Clinic mentors who supervised and mentored the students in their research labs this summer, and especially those who helped our students get the extra experiences that were really value added. Thank you to the Interdisciplinary Health Sciences Institute, who administratively supports the Mayo Clinic and Illinois Alliance and the SURF program on the Illinois campus. And thanks to all of you for being here today to help to support our Mayo Clinic SURF fellows. And an overwhelming thank you to our 2021 SURF Fellows for sharing their research experiences, their photos, and their videos from the summer. It was really great to get those from you all summer long. So thank you. And we wish each of you the best in your future endeavors. So thank you, everyone.